Hello, dear students. In this lecture, we will discuss microchemical tests done in economic botany laboratory. So, the first test which we are going to discuss is starch test. To test the presence of starch, we require uh, a test solution. Then we require test tubes, holder, spirit lamp, iodine, hydrochloric acid, Benedict solution, NaOH, etc. So for this we have two procedures. First is you just take a pinch of starch and add some water and add a few drops of iodine, so the blue black color appears. And the second method is that you take a pinch of starch, add some water, boil it by adding some HCl, and make the solution alkaline by NaOH or sodium carbonate, and then test with Benedict solution, so the red precipitate is formed. So if we can apply any of the tests, but usually we do the first method, which is very simple, by taking starch and taking adding iodine. So the test solution is taken and iodine is added. If the blue black color appears, then we say that the test is positive for starch. So here you can see in the first two pictures that first test tube is control and the second is with test solution. So there is no blue black color in control, while in test solution the blue black color appears. So we say that starch is present in test solution. Now we can also see starch grains under microscope. So for this, you just take some paste of test solution, put some iodine on slide, put cover slip and observe under microscope. So this iodine stained starch grains appear blue when they are viewed under microscopes, and we see different types of starch grains depending upon what plant we are taking. And we can see eccentric or concentric starch grains under microscope. For example, here in the first picture we can see the concentric starch grains in wheat, and in maize starch grains we see that it is somewhat different from wheat because it has a starry shaped starch grain. Now, the second test which we are discussing is the test for reducing and non-reducing sugars. So, reducing sugars are monosaccharides, which contains aldehyde group, and they are known as aldolysis, while those which contain keto group like fructose are ketoses. So these sugars with a free or potentially free aldehyde or keto groups are thus known as reducing sugars. And there are two tests for to test reducing sugars: Felling's test and Benedict's test. And then non-reducing sugars, we do hydrolysis test for non-reducing sugars. So let us discuss these two tests. So like first test is for reducing sugars. So for this we have two procedures: Felling's test and Benedict's test. So for Felling's test we take um, some test solution and add Felling solution in test tube, and then we boil it, and we see brownish red precipitate. Then we see that it is positive. For Benedict's test, we take some test solution, add Benedict's solution, and then we boil, and we see, if we see red, reddish yellow or green precipitate, we say that the test is positive. So here you can see the results of Benedict's test and Felling's test in test solution. We see that the first picture is showing very feeble presence of reducing sugar because This is Benedict's test, and we can see that it is greenish. So this is very small amount of reducing sugar is present. Similarly, in second picture also, we can see that the green precipitate has formed, so the test is positive. While the third picture at the bottom shows Felling's test positive, having brick red PPT. So these are the results of reducing sugar, which we see, and what conclusion we derive that sugar, when it is treated with alkaline solution, it undergoes enolization to produce enodiols, and these are highly reactive reducing agents which are capable of reducing oxidizing Cu3 2 plus ions. So both Fellings and Benedict solutions contain soluble Cu2 plus ions in soluble form, which forms a complex with citrate or tartrate. And when they come in contact with any diols Cu2 plus, which is cupric, and they are reduced to cuprous ions, which is Cu plus ions, and they later combine to precipitate yellow cuprous hydroxide. So yellow PPT of cuprous hydroxide on heating is converted to reddish cuprous oxide, and this is the principle of the reducing sugar test. So you can see the reaction says that when sugar or monosaccharides they undergo, when they are treated with alkali, they convert in anidiols, and Cu2 plus is converted to Cu plus. So this gives the color. So here the overall reaction, like glucose is a monosaccharide, which in the presence of this Cu2 plus ions, when it is boiled, it gives rise to um, gluconic acid and cuprous oxide. Now, the next test is for non-reducing sugars. So here, the non-reducing sugar, the example of non-reducing sugar is sucrose or starch. So, if for this, the procedure is that you add um, sugar solution, or you add the test solution, which is in equal volume to the concentrated HCl, then boil the mixture for about 5 minutes. During this time, the hydrolysis will take place. Then you neutralize the resulting solution with sodium carbonate or bicarbonate, and then you subject this solution for the test of reducing sugars by adding Fellings or Benedict's, anyone of your choice. So, what is the result? If the result for Fellings test it gives red PPT, and for Benedict's test you get red, green, or yellow PPT. So, what does that mean? For this, if we are getting negative result in when we test for reducing sugars, then we if we are testing for Fellings test and Benedict's test, and we are getting negative results, so we hydrolyze them and again test for them, and then if we get positive, we see that non-reducing sugar is present. So what is the conclusion? This test reveals that the presence of non-reducing sugar, and the non-reducing sugar is sucrose, which occurs widely in plants. It is basically present in sugarcane. So it is composed of glucose and fructose, and when this they are joined together by glycosidic bonds. So when hydrolysis of sucrose takes place, it is hydrolyzed to glucose and fructose and gives the test positive for Benedict's or Fellings test. Now the next test is for proteins. For proteins we have three tests: xanthoprotic test, biuret test, and Millen's test. So in xanthoprotic test, we take the test solution and add a few drops of nitric acid. A cloudy white precipitate is formed, which turns yellow on warming. When we cool it and add ammonium hydroxide, To form make it basic, the color changes to orange. In biuret test, the test solution or the protein when the protein solution is added with dilute solution of caustic soda and we add few drops of copper sulfate solution, a bluish violet color is produced. In Millen's test, we add few drops of Millen's reagent to the test tube and it contains few ml of protein solution. Then a white precipitate is formed, which on boiling turns reddish brown, and the color deepens when we add NaNO2. So, the Millen's reagent is prepared by adding one part of mercury to one part of strong nitric acid, and it is also commercially available with the agents, so we can directly buy them. So here you can see the results of xanthoprotic test and biuret test done for protein presence. And here in biuret test you can see that the first test is is blue. So this bluish color shows the presence of proteins, and the xanthoprotic test you see that the color has changed to orangish yellow. So here we say that it is the protein is present. So what is our conclusion? The color changes indicate that the presence of protein is there, and biuret, it gives violet color when it is treated with dilute CuSO4. So this reaction is shown by compounds having CONH2 groups which are joined by C or N atoms, and proteins all gives this reaction because they possess a CONH peptide bond architecture. So and in xanthoprotic reaction, nitration of phenyl ring occurs to yield yellow substitution of products which turn orange on addition of alkali. So here you can see the results of xanthoprotic and biuret test. So here you can see the results of biuret test, Millen's reagent, xanthoprotic test. Now the last test is for oils and fats. So for this we take the test solution and we add alcoholic solution of Sudan 3 or Sudan 4, and the oil will be stained red. So here you see that when this is the test showing Sudan test for fats. So you see that this it has turned red in color, and we say that the test solution has fat present in it. So what is the first method to test it? We cut some sections of the material, place the section in Sudan 3 for about 10 minutes, and then we wash with alcohol and observe under microscope. So we see that um, um, oil drops, they take a red stain. And in the second method, we take a few drops of osmic acid to the test solution, and if black color is developed, we see that fat is present. So here you can see all the tests in wheat, like in the first test is Benedict's test, second is Benedict's test for reducing sugar, Millen's test for proteins, iodine test for starch, and Sudan test for fat. So you can see how the test solution is compared with the control solution. And here that these are the tests for maize. So you can see blue color in starch. Then you have different colors for Benedict's hydrolysis test for non-reducing sugar in maize. And these are some more pictures to show iodine test for starch, reducing sugar test, Benedict's for proteins, biuret test, and fat Sudan test. So this is how you can represent your results when you compare different plants, and you can also show them individually. So thank you, students. Let us meet in next lecture.